Angiogenesis is the complex process of new blood vessel growth, which supplies oxygen and nutrients to support tumor expansion. Key players in the control of angiogenesis are receptor tyrosine kinases and their associated growth factor ligands. These kinases represent a promising group of molecular targets for anti-cancer drug discovery. When a tumor outgrows its blood supply and becomes hypoxic, the oxygen-starved tumor cells call for nutrients by releasing a number of angiogenic growth factors. Vascular endothelial growth factor, or VEGF, and basic fibroblast growth factor, or BFGF, diffuse into the nearby tissue. VEGF binds to and activates specific receptor tyrosine kinases on endothelial cells. VEGF initiates blood vessel assembly by causing vascular endothelial cells to migrate, proliferate, and assemble into new, although poorly organized blood vessels. VEGF also protects the immature blood vessels from apoptosis. Platelet-derived growth factor, or PDGF, assembles smooth muscle cells and parasites in an attempt to stabilize and support the new blood vessels, and also protects against apoptosis. Now attached to its own blood supply, the tumor is able to grow and spread. Both the new abnormal and poorly constructed and the more established tumor blood vessels are subject to selective therapeutic intervention. This pathological process can be reversed even after the abnormal blood vessels are established. The VEGF, BFGF, and PDGF receptor signaling can be inhibited using small molecule tyrosine kinase inhibitors, which work intracellularly. These receptor tyrosine kinase inhibitors easily cross the cell membrane and bind to the adenosine triphosphate ATP binding site of the intracellular kinase domain of the receptor. Growth factors may still bind their receptors, but the receptors are blocked from signaling for cell migration, proliferation, or survival by the tyrosine kinase inhibitors. Without activated growth factor receptors to protect the blood vessels, the cells making up the abnormal blood vessel undergo apoptosis, and the blood vessel degrades. This stops further tumor growth and may result in necrosis of larger